So today our speaker is Professor Jose Nelson, and he is a professor at University of California, Los Angeles, which we usually abbreviate UCLA for short. So I met Hosea not in person, Dr. Nelson, not in person, but through this magazine. Some of you in my class, in this class, should know what this is. Chemical and Engineering News, okay? It's the weekly news magazine for the American Chemical Society, which is the largest professional scientific society in the world, okay? So anyway, they started a feature called the Talented 12, um, a rigorous selection process, 12 young professors, young entrepreneurs who are making big impact already in their careers using chemistry to solve global problems. And featured on page 26 is our speaker today. That's where I saw him. And I said, "Oh, yeah, we got to get him over here." So that's how this all started. In addition to the fact that he is, like myself, an organic chemist, he also has a very interesting, at least educational, he probably also has a very interesting life story, but educational story in which he originally thought about getting into the construction business as a sheet metal worker. Then he took a class. I guess that wasn't going too well or something. Then he took a class at a community college, a chemistry class, and that basically kind of changed the trajectory of his educational career. And now he is a first year, hasn't taught a class yet, right? At UCLA. So it's a very astonishing and fascinating personal story to go along with his expertise in the area of organic synthesis. So it's a pleasure to have you here. It's an honor to have you here, and we look forward to your presentation. Yeah, I don't think we could hear you. I don't know if you could hear us. He's talking.
How about now?
Okay, there you go.
Okay,
Did you
okay.
hear the introduction?
Yes, I did. It was a fantastic
Oh,
introduction, and I've already
oh,
thanked you so many
I
times
just
for
okay.
it. But
Okay.
what I wanted to say is I wanted to thank you for the kind introduction. And one thing is I'm about to bombard you, or maybe not bombard you, but talk a lot of hardcore organic chemistry. But before I get into that, I will acknowledge that my background is different. And I think anytime I go to an institution that is a primarily undergraduate institution, I do my best to tell people that you can always change and you can always dictate where you're going in your life, no matter what point of your life you're at, if that makes any sense.
Yeah, that sounds pretty cool. You know, we have your audience today. All of them, or most of them, are eating ice cream.
Hmm.
Just so you know, it's kind of relaxed back here. So you bring
Okay.
on the chemistry.
Good. And you guys should have sent me some dry ice packed ice cream so I could join you. But today I'm gonna tell you guys about a lot of the work that I think I don't think I know got me to where I'm at today, and it really encompasses both a 5-year PhD and a 3-year a stint as a postdoctoral scholar. And, I chose to organize the work that I'm going to tell you about today because I think it's representative of two major paradigms in organic synthesis. And so I guess the title says, strategies to prepare complex molecules. And it outlines, it says target-driven synthesis and reactivity-driven methodology. And the two stories I'll tell you today are fall in one of those categories. But I think the take-home message for those of you who are thinking about pursuing research careers in organic synthesis is that both of these pathways and both of these strategies in research can lead you to the preparation of fairly complex molecules that are biologically relevant. And so in the first part of the talk, I'll tell you about my work in total synthesis that I undertook during my PhD with Brian Stoltz at Caltech. And in the second part of the talk, I'll tell you about my work within the area of chiral anion catalysis with Professor Dean Tost at UC Berkeley. And the story of the natural products that I worked on begins with this plant shown here on the left. This is a plant called Thapsia garganica, and it's native to the Mediterranean and northern regions of Africa. And it has a really rich history. And so Hippocrates, well before the birth of Christ, wrote about its medicinal qualities, how it could be used to treat pulmonary disease, runny nose, rheumatoid arthritis. It also has, I think, some more infamous historical references. So the Emperor Nero frequently used paste derived from Thapsia plants to treat blemishes and bruising. And it also has this nickname, the stinking carrot or the deadly carrot. And you can see this kind of old, several hundred-year-old drawing up here in the middle of the screen that has some Old English, but it says deadly carrots. And the reason why is because the roots of Thapsia plants look like carrots, and people traveling through the arid regions of North Africa would occasionally stop, dig up a Thapsia plant, see that the root looked like a carrot, and eat it, and then die. And so this is where that name came from. And so it contains very cytotoxic and very toxic compounds. And fast forwarding to modern times, what we figured out is that this compound shown in the bottom left-hand corner, thapsigargan, is responsible for much of the cytotoxicity or much of the toxicity of plants belonging to the Thapsia genus. And so this toxicity has been utilized to make drugs, and so people are now using thapsigargan-like molecules. There's a couple compounds that are in clinical trials to treat prostate cancer, which use this molecule or molecules like it to achieve that, to kill cancer cells. But I think thapsigargan is more well known for chemical biologists and cell physiologists using it to probe calcium homeostasis within the context of a variety of cellular studies. And so thapsigargan is a SERCA inhibitor. And so these SERCA enzymes are these big complex enzyme complexes that sit on the surface of the endoplasmic reticulum and will pump calcium ions into the cytosol, and that process is driven by ATP. And so when you make molecules or have molecules that inhibit that, what happens is calcium concentrations build up in cells, and the cells will die. They'll undergo apoptosis, and I think in this case it's purely from osmotic pressure of having such high calcium concentrations. And so what cell physiologists have started doing, I guess around 30 years ago, was using thapsigargan as a probe molecule to look at calcium homeostasis. So if they're interested in understanding the effect of calcium ions on some process, what they can do is use thapsigargan to change that in an in vivo study. And so to these ends, it's been utilized a lot in the literature, referenced a lot. So I would say, I guess this is a typo. This is not 142,000, but it's mentioned in 14,000 publications since its isolation, which is more than one a day. You can buy the compound for around $100 a milligram from Sigma Aldrich or from Invitrogen. And so due to this really powerful application, isolation chemists have spent a lot of time in plants belonging to the genus Thapsia looking for structural analogs of thapsigargan that have better potency, or perhaps that they can isolate in greater yield. And so in the context of one of these studies, Massenet and Appendino isolated these compounds shown on the right, which they termed the transdagonalides and bacillialides. And I think most of you guys in the audience, kind of regardless of where you are in your chemical education, can recognize that the shapes of the transdagonalides and bacillialides is very distinct from thapsigargan. In fact, I'm hard-pressed to think about any real substantive structural correlation between these two classes of compounds. And what was amazing to the isolation chemists, and I think amazing to us, is in our initial query into making these compounds, is that despite these structural differences, despite the fact that they're completely different, these compounds actually do the same thing. And so we thought that was very interesting, and perhaps a neat example of convergent evolution, and interesting from a philosophical level. These plants actually make these things that we use as drug molecules for a reason. They also have something that they're trying to treat on the molecular level. And so thapsigargan itself, its molecular target is well known. So it actually fits right into this transmembrane portion of the circa complex, and that's where the inhibition comes from. Transagonalide C, on the other hand, we don't know how it works. I won't go into it today, but there's a considerable amount of evidence that it works via a different mechanistic pathway. And so I think one of the things is that transagonalide C is isolated in very small quantities. So if you take around 20 pounds of thapsia root and grind it down and do an extraction of the chemical components, you'll get a few hundred milligrams of thapsigargan, and much greater quantity. Transagonalide C, for example, I think in the isolation papers I've read, is around 2 or 3 milligrams of compound out. So, with an eye towards these biological activities, we wanted to synthesize these compounds to, perhaps make compounds that were applicable to human health and perhaps even to fundamental biological studies. But in the end, we're synthetic organic chemists, and I think what excites us even more than biology is unique structural elements. And so if you look at the transagonalides and bacilulides, they have a number of structural features that are pretty interesting. At least I think they are. And so you can start by looking at this fused AD ring system, which I'm highlighting here in transagonalide C and D. And this type of 2,2,2 oxobicyclooctene is known in the synthetic literature. People have made compounds like this, but they're known to be thermally unstable. They decompose via Retro-Diels-Alder process to release CO2. Furthermore, this C ring, this oxapinone, or what we call a cyclic carboxyketene acetal, for those of you who have done some synthetic chemistry, this type of functional group, this alkene bound to an oxygen bound to a carbonyl, is actually a very unstable group. So it's very sensitive to water. And so these compounds are actually derived from water. They come from a presumably aqueous physiological environment. And so the fact that this type of structural element exists in a naturally occurring compound is also interesting. There has to be some element of stability that comes from these, the whole of this molecule, as opposed to just that one functional group. In a more traditional sense, the compounds have a considerable amount of stereochemical complexity. So, for example, baciluliide C has 6 contiguous stereocenters, 2 of which are all carbon quaternary centers, I guess the C4 and C8 carbons. Also in the family are these compounds, transagonalides A and B. So these are considered by many to be the most complex members of the family. Replacing that 2,2,2 oxobicyclooctene moiety is this fused gamma-butyrolactone, which is bridged by an ether linkage, forming this complex ADE tricyclic core. These compounds have up to 7 contiguous stereocenters, 2 of them being all carbon quaternary stereocenters at C4 and at C8. And so taking some of these structural elements into consideration, we devised the following retrosynthetic hypothesis. What we envisioned, and largely due to the perceived instability of this type of carboxyketene acetal, is that we would form it at a late stage in the total synthesis, perhaps even in the last step, from a tricyclic vinyl halide, such as this compound shown here. In turn, recognizing that 2,2,2 oxobicyclooctenes are often formed via Diels-Alder reactions, we envisioned that this precursor could be formed via Diels-Alder cyclo addition of this type of pyrone with a tethered dienophile. And finally, recognizing that this C8 quaternary center, alpha to a carboxylic acid, could be derived from an Ireland Claisen rearrangement, we envisioned that the construction of this type of pyrone could lead directly to our Diels-Alder cycloaddition precursor. And finally, and really importantly within the context of our total synthesis, we thought that we can access all members of the natural product family by simple variation of this R2 and R1 groups on these Draniol derivatives, many of which are already known and reported in the literature. And so the first element or the first challenge associated with this retrosynthetic hypothesis is this Diels-Alder cyclo addition reaction. As I mentioned before, there's a considerable amount of literature precedent that says that these types of systems, these 2,2,2-oxobicyclooctenes undergo decarboxylation. And so we built a simple system, just this pyrone with a tethered dienophile. And what we found is that it took heating to 120 degrees Celsius to affect a cyclo addition reaction. Unfortunately, however, at this elevated temperature, this intermediate 2,2,2-oxobicyclooctene was not stable and would let loose CO2 in a retro-Diels-Alder process to make this undesired diene. And so, guided by a lot of key work by Gary Posner and others, what we found is that halogenation of this pyrone ring lowered the activation energy of the cyclo addition process to 80 degrees, such that we were able to isolate this 2,2,2-oxobicyclooctene in good yield, which really represents the core of the compounds we're trying to make. And so our next step was to affect this Claisen rearrangement, which is highlighted here. And so we were actually quite pleased to find that a simple modification of those conditions initially disclosed by Bob Ireland back in the 80s allowed us to transform this bromide into a precursor via an Claisen rearrangement that we thought would be suitable to undergo our pyrone Diels-Alder reaction to build the core. And so as soon as we had this compound in hand, we tried using our conditions that we had developed. We had the bromine in place, so we thought we could do it at lower temperatures. But what we found is that this compound would undergo non-productive decomposition and didn't make this tricyclic carboxylic acid. Instead, what we saw was decarboxylation at this C9 position. And so what we reasoned was that the intermediate immediately following the Claisen rearrangement is actually a carboxylate, or it's actually a silyl carboxylate, probably didn't undergo the decarboxylation. It had to be hydrolyzed and protonated and decarboxylate from the carboxylic acid moiety. And so we envisioned that if we could do the chemistry very carefully without introduction of water or air, we might be able to get this cyclo addition reaction to occur. And so basically, with very careful Schlenkline technique, we did our Claisen rearrangement to get to this intermediate silyl ester, and then we took off all the solvent, took off all the THF solvent, replaced it with benzene, and then heated this reaction mixture for 13 days, and we're able to achieve a facile synthesis of the core transagonalite C and D. Ultimately, after a significant amount of optimization, we're able to take this pyrone iodide and treat it with BTSA, which is bis-trimethylsilylacetamide, at elevated temperature for 6 days to directly form in a shorter time period the tricyclic core of the natural product. And so for those of you considering going to grad school, at the end of your PhD or master's, you may give a talk like this one. And so I'm looking at this slide, which is just one slide, but it really represents around 2.5 years of my life. But in the end, it was very difficult to make this C-ring, and we explored a lot of different strategies to achieve this annulation. What we ended up having to do was use this type of metylated methoxyacetylene, do a cross-coupling reaction to make this type of E9, which is not stable at all, but then this undergoes cyclization to make the C-ring of the natural product. And so the way this looks in the complete version is that we could take our iodide, we can expose it to our Claisen Diels-Alder cascade rearrangement conditions to make this tricycle. Then we protect this carboxylic acid as the TBS ester. Then we do a cross-coupling reaction to make an intermediate E9. This E9 is not stable, but when we expose it to physiological conditions, it immediately cyclizes to make these diastereomeric mixture of transagonalite natural products. Overall, this total synthesis is 7 steps from commercial materials, so things you can buy from Sigma Aldrich or another vendor. We use one protecting group, just this step right here where we put in this silyl ester, and the penultimate intermediate is available on gram scale. And so we thought this was a general strategy to synthesize these compounds. We went ahead and made a number of the members of the family using this strategy just by simple variation of this R group. So moving on, what we needed to do was achieve this same synthesis. However, we needed to do it in an antioselective fashion. And so we chose to explore this within the context of transagonalides A and B, which are the most complex members of the family. And what we thought is, just like transagonalide C and D, we would form this 7-membered ring late in the synthesis via what we call the formal 5+2 annulation of this type of iodide. Importantly, we thought that we could form the tetracyclic core of these compounds via acid-catalyzed translactonization process of this type of aldehyde. And so the way we thought this would occur is under aqueous acidic conditions, we would hydrate this aldehyde, and it could undergo an acid-catalyzed translactonization to make this gamma-hydroxybutyrolactone. And in under acidic conditions, this could eliminate out water and undergo a transannular cyclization reaction to make the core of the compounds. And so that was part of the challenge, just constructing the core of these compounds, but also we had to think about ways to do this enantioselective Ireland-Claisen Diels-Alder cascade reaction. And so there's a number of different substrates you could think that we could employ within our Ireland-Claisen Diels-Alder cascade. You could think about using this type of aldehyde. However, we knew from some of our studies in racemic systems that was not competent under the reaction conditions. We also knew that we had problems with protected alcohols because we could have used this and oxidized it later, the aldehyde. And so what we were left with is trying to use this Z-enoate, having that undergo the Ireland-Claisen Diels-Alder cascade, and then doing a selective reduction of the methyl ester, shown here with in the green, to the aldehyde. And there's a number of challenges associated with that. So reductions of methyl esters to aldehydes, certainly a reaction many people will learn about in a sophomore organic class, but in practice, it's a very challenging reaction with very low levels of success. And there's a lot of things associated with it practically. Usually it's a dibal is your reducing agent. It's very low temperature, weakly coordinating solvents such as hexanes or toluene, and there's a lot of criteria that have to all kind of fall in line for this reaction to work. And so in this complex substrate, we were very, challenged. It was a somewhat risky approach, and for a number of reasons. And the first reason is that this type of carboxylic acid would probably or almost certainly be initially deprotonated by our reducing agent. And at the low temperature we wanted to do this type of chemistry that may render our substrate insoluble and unreactive. The second thing is this bridging lactone, the second carbonyl shown in red, we had found in our hydrolysis studies that was at least with respect to hydrolysis, the most reactive carbonyl in the molecule. And then finally, and the greatest challenge was that the desired site of reduction, this carbon shown in green, is appended to an all-carbon quaternary center. And it turns out that, we thought that kinetic restriction from that all-carbon quaternary center may provide an insurmountable kinetic barrier to achieving this reaction at the low temperatures required to get a single hydride reduction. But nonetheless, it was kind of the only option we had available at the time after a lot of experimentation. And so we were able to prepare this chiral geraniol equivalent using a method first described by Hoppe and coworkers in 2005. After a five-step sequence, we're able to advance that to the Z-enoate, which underwent smooth esterification with this pyrone ester to make the substrate for our Ireland-Claisen Diels-Alder cascade reaction. And so we were very happy to find that exposure of this substrate to our Claisen Diels-Alder cascade conditions did result in formation of this diastereomeric mixture of products. And furthermore, the chirality of this starting material, this monocyclic starting material, which was 90% E, was transferred with high fidelity into this diastereomeric mixture of products. And so we subjected this to a number of conditions. And in the end, what we found is that if we use 5 equivalents of dibal as a reducing agent, we're able to form this aldehyde in very high yield. And furthermore, we were ecstatic to see that treatment of this compound with aqueous HbF4 led to cleavage of this vinyl silane and also concomitant structural rearrangement of the core of the molecule to make this tetracyclic natural product core. So we applied our 5+2 annulation methodology, namely we protected the carboxylic acid as the TBS ester, and then we did a Stille-like cross-coupling to make an intermediate anion, and once again, upon exposure to physiological conditions, this cyclized to make mixtures of transagonalides A and B. And so in this first part of the talk, I described to you a rapid total synthesis of transdiagonalides A through D. We also made bacilliolides A through C using that methodology. We developed an enantioselective Ireland Claisen rearrangement Diels-Alder cascade, which allowed us to prepare all these molecules with high levels of enantio enrichment. And I didn't have a chance to talk about it today, but any of you guys who are interested should check out some of the references that I had on my slides, largely because a lot of the work that we did in this area was contributed to some of our knowledge of the biosynthetic origin of these compounds, which is actually pretty interesting. So that represents the work that I did during my PhD studies, and really, as I alluded to in the beginning of the talk, it's a prime example of target-driven organic synthesis. You pick a target such as transdiagonalide A, for example, and then you. I don't want to say by any means necessary because that may have a negative connotation, but really, I mean, however you can, you figure out a way to make transdiagonalide A. And you know, in our case, we had to develop some new methodology and do things, but the end result, having that compound in a flask that we synthesized, was what was valuable. So I wanted to expand my training, and as I mentioned before, there's another paradigm within the field of organic synthesis where people look at reactivity-driven organic synthesis. So they'll have some sort of reactivity they're interested in, and then they'll use that reactivity to make a lot of different molecules. And they're less concerned with the molecule they make in the end than they are with how they made it. And so Dean Toss from UC Berkeley is certainly very well known for using this paradigm in research. And so he's uses the pi acidity of gold 1 to do a lot of interesting transformations, and also uses the concept of chiral anions to do a lot of interesting transformations. And shown here is a catalyst that bridges those two concepts, a cationic gold 1 species with a chiral anion associated with it. And this is really where I got excited. I really wanted to join Dean's group or Professor Toss' group to learn about how to control enantioselectivity in transition metal catalyzed reactions using chiral anions, such as this molecule called TRIP. And so I think that's important because I think in a lot of regards, it represents a completely independent way of thinking about asymmetric catalysis. So traditionally, the way that asymmetric catalysis is carried out, at least with transition metals, is that we have some catalyst that's derived from a reactive metal center, which is ligated to a chiral organic group. And this acts upon a prochiral starting material substrate to make enantio-enriched chiral products, where the chirality of the product is derived or transduced from the chirality of this organic group that's on the metal. And so this strategy was disclosed by Knowles, Nauri, Sharpless, been worked on and advanced by a number of people within the field, and I think has been a driving force for organic methodology for around 30 years, and a Nobel Prize was awarded in 2001. And here's an example of the strategy where you could take this alpha-beta unsaturated carboxylate and using a rhodium metal center ligated by this chiral bisphosphine, make highly enantio-enriched L-dopa precursor. So it's a very important area, and so it would always be good to have alternative strategies to do asymmetric transition metal catalysis. And so one way that people have thought about doing this was to use a different approach, where you had a cationic metal and an anionic chiral counter anion, or a chiral X ligand, and the chirality that you would get in a transformation going from a prochiral starting material to an enantio-enriched product, the chirality would be derived from this chiral X ligand. And so people such as Barry Trost, Arnstead, Milstein, and a number of folks have tried in the 90s and early 2000s to achieve this, and they had some moderate levels of success with some low levels of enantioselectivity. In 2007, Professor Tost gave the first example of a highly enantioselective chiral anion mediated transition metal catalyzed process, this cyclization of these types of allenes, which are able to proceed in 97% E and 90% yield using this TRIP molecule, this anion shown in the bottom right-hand corner. And in a lot of regards, the Tost group was very lucky because there's some unique and subtle mechanistic features of cationic gold catalysis that makes it exceedingly well suited to have made this discovery. And so I think a really important element of this is that in a catalytic cycle, perhaps for the reaction I have drawn on the top of the screen. Oops, sorry. Jump ahead. This cationic gold species is preformed with a chiral anion, and for the most part, throughout the catalytic cycle, there's always either a cationic gold or there's a cationic substrate associated with it. And so the chiral anion could stick around and be turned over, and so you can use catalytic amounts of it. But transition metal catalysis is a pretty big field, and actually, in the scheme of things, gold catalysis is not necessarily the most commonly employed transition metal. If you think about something like palladium that people use all the time in the pharmaceutical industry and in academic research, how do you think about applying this chiral anion approach to a palladium catalyzed reaction? And so shown here, for example, is a prototypical, I guess this is a Heck-Matsuda arylation reaction. And so in this type of scenario, there are some challenges associated with using chiral anions. And so perhaps the biggest challenge is this, is that if, let's say that you're able to take this aryl diazonium cation and you are able by some method to associate it with the chiral anion. This can undergo an oxidative addition process to make this palladium-2 cation with an associated chiral anion. So that chirality of the anion is still associated with the reactive metal, and we're good up to this point. Now you can do the migratory insertion reaction, which is classically, in a lot of regards, the enantiodetermining step. And here you've generated your chiral molecule, essentially. You still have your chiral anion associated. This can beta hydride eliminate to make your enantioenriched product. And you have your palladium-2 cation here with your chiral X ligand, so everything is good. However, in a key step, a key fundamental organometallic transformation in a Heck reaction, for example, problems arise because you have to go from this palladium-2, this cationic palladium-2, to a non-cationic palladium-0. And this usually occurs through a reductive elimination process. And in this case, for this example I have drawn here, what you'd end up doing is precipitating out your chiral anion. And so you'd have to use a stoichiometric amount of this very extensive chiral anion to do asymmetric catalysis, at least traditionally, at least up to the point where we started working on this. And so really, the game here is, how do you get this chiral anion out of this salt, which is probably precipitated out in the bottom of your flask? How do you get that back into solution and reacting? And so right about the time we started to think about this pretty big problem, other members of the TOS group had developed some of this chiral anion phase transfer methodology. So to be clear, this is a solid liquid phase transfer process. So the way this works is you'll have some very soluble lipophilic chiral anion. You can have a very insoluble cationic reagent, and when you mix those two together, the chiral anion will undergo a salt metathesis reaction to make a very soluble chiral ion pair. So this essentially, your chiral anion pulls your cationic reagent out of solvent. And then this soluble chiral ion pair can undergo an enantioselective reaction with a prochiral substrate to give you an enantioenriched product. And so a classic example here is using this molecule here, which is called Select4, which is very insoluble in anything but, say, water and acetonitrile. And when you use a chiral anion, such as those derived from binol, you're able to make this salt, which is very soluble in many solvents, including benzene and toluene and more nonpolar solvents. And you can use this to do asymmetric fluorination. So the idea was can we use these phase transfer reagents with something like an aryl diazonium salt, which is also insoluble, to make this type of soluble chiral ion pair, which could then be used in an enantioselective Heck reaction, for example. And so the way this looks in the scheme that I showed you earlier, the way we would turn this over is that you would have an insoluble aryl diazonium salt. This could undergo a chiral anion phase transfer process, which would lead to a soluble chiral ion pair. So X is the chiral anion. This could undergo oxidative addition to generate this palladium-2 aryl cation with an associated chiral anion, which would then undergo migratory insertion to make this a cationic palladium sigma complex. This would be the enantiodetermining step. Beta hydride elimination would yield your enantioenriched product, and now your palladium-2 cation with an associated chiral anion would indeed undergo reductive elimination to make a non-cationic palladium-0, which can associate a chiral anion. But what that would do is that would also regenerate a phase transfer catalyst. And this can go on and resolubilize your insoluble aryl diazonium salt. And so this is how we envision we could overcome a fairly significant barrier of doing chiral anion catalysis with a palladium 0 type catalyst. And so we were very excited to find in our initial efforts that we could aerolate dihydrofuran with this toluene aryl diazonium salt, and with some levels of enantioselectivity, so 57% EE, and at a lower conversion using this TRIP, which is this bienol-derived phosphate in diethyl ether solvent, and with this inorganic base in palladium DBA. And so these numbers, for those of you who follow the field of asymmetric catalysis, know that these numbers are, while they're very encouraging initial results, they're not necessarily game changers in the field. And so we spent a lot of time trying to optimize this process. And if any of you guys go on to do some ion pairing catalysis, or what you'll find is that it's very tricky. So solvent polarity, for example, has a tremendous effect on enantioselectivity and conversion in these systems. And so a lot of times, at least in the context of this project, we started to look at substrate optimizations. And so what we found is that electron-poor alkenes, such as this alpha-beta unsaturated ester, had no reaction, and we had some poor reactivity, at least at this point, with these times electronically unbiased alkenes. And as you see in this example above, moderate conversion with dihydrofuran. And actually, when we started to go to more electron-rich alkenes, we had complex mixtures of products. And I'll come back to that later. In an ideal sense, from a reactivity perspective, it'd be very nice if we could do an enantioselective Heck chemistry on an alpha olefin, such as this compound drawn on the left. However, unfortunately, these Heck reactions undergo beta hydride elimination to make styrene, so they don't generate a stereocenter, so you can't do them asymmetrically. However, we were very excited to see that we had very good conversion and yield in these types of systems using our chiral anion phase transfer process. So what the question became was, how do we manipulate this system, which canonically gives achiral products, and make it give enantioselective products? And so right around the time we were looking at this, Matt Sigman from Utah had found that you could take alpha olefins, such as this protected allylic alcohol, and under a similar set of conditions generate these diarylated 1,1 diarylated alkanes. And he didn't do it enantioselectively, but he did make something that was chiral. And so we thought this was a very good starting point for us to perhaps exploit this very good reactivity with acyclic alkenes. And so what we thought is we could use this, right? And the way this works from a mechanistic standpoint is you make some sort of palladium 2 aryl cation, which could undergo Heck insertion to make this palladium 2 cationic sigma complex. And then this can undergo beta hydride elimination, followed by subsequent reinsertion to make this benzylic palladium cation with an associated chiral anion. And then a nucleophile could come in and attack at that one position to make this 1,1 disubstituted a substrate. And so we looked at a lot of different nucleophiles, boronic acids, aryl stannanes, carbanion type nucleophiles, and really, for the most part, these reactions lead to multitudes of products. You have, in some cases, we'd see some formation of our 1,1 substituted chiral product, but a lot of times we just see Heck olefination, or we would see just quenching of the diazonium cation with the nucleophile. And so we were really excited when we got to diboron reagents, because what we found is that with diboron reagents, and this is unclear exactly why at this point, we saw very clean formation of 1,1 difunctionalized compounds. And so we decided, before we even looked at enantioselectivity in the system, to examine the racemic scope. And so what we found is that protected allylic alcohols, homoallylic alcohols, bis-homoallylic alcohols all undergo smooth aryl borylation. Allylic amines undergo aryl borylation. Astonishingly, we can take electron-poor alkenes such as acrolein, vinyl ketones, acrylates. These all undergo aryl borylation. We can even take 1-octene, and that undergoes aryl borylation. So it's a very general reaction we discovered. Very broad reaction. And so I think this goes to the main point of the talk is, within the context of our research, we're really just looking for ways to use chiral anions with non-cationic metals. But as shown in this slide, we stumbled upon a really powerful transformation and a really powerful way to make complex molecules. So, at almost concurrently, time-wise, while we're doing this chemistry, there's all these other groups publishing really nice papers on doing aryl borylation. So Hall, Yu, and Morkin, they can take substrates and do aryl borylations and make things very similar to what we're making, but usually in, say, 6 steps with 2 transition metals, 2 complex phosphine ligands, complex boron protecting groups. And so what we're doing is doing it in 1 step. So we spent a good amount of time, re-optimizing our asymmetric or enantioselective conditions. And what we found is that we're able to do this aryl borylation reaction on with high levels of enantioselectivity, pretty good yields on systems as vast as protected allylic alcohols and also acrylates with a very similar set of conditions. A key development or a key discovery in enabling this chemistry to work was finding that we had to use exogenous diene ligands. So this is meta CF3 DBA. And so what we found is that dienes in general as additives were very effective in increasing the yields of these reactions. In particular, this meta CF3 DBA increased the yield of the reaction and did not harm the enantioselectivity. So having kind of provided proof of principle for using chiral anions with say, palladium 0 species, we've expanded this to a number of methodologies, some of which I have on my slides, some that you'll have to read about. But we're able to take dienes, and then these compounds undergo an interesting Heck process, followed by quenching with an internal nucleophile to make benzopyrans. We can make benzofurans with good enantioselectivity. We can take these types of Tazel-Bach amines, or these cyclopentenes, and we can do enantioselective arylation. We have a good number of examples with good numbers of enantioselectivity. We're actually going to submit a paper pretty soon in this area. And so I guess before I finish up this part of the talk, it was, I had a slide up earlier where I said, as we went down the electron the electronics of alkenes, we got more electron rich, and we started to see mixtures of products right when we started to get to these type of proles. And we went farther, actually, and we looked at indoles, for example, this scatol molecule. And so what we found is, under these conditions, we did see quantitative yield of a product. And we actually, initially thought that we'd C3 arylated scatol. But after we heard back from the mass spec facility, we found that we, at least according to them, that we still had dinitrogen incorporated into our molecule. And so we looked for some other indole substrates where we might test to get final structural confirmation, hopefully unambiguously via X-ray crystallography, to see what we were actually making. And so what we envisioned is that we could take this type of tryptamine, and then this could attack the terminal nitrogen of an aryl diazonium salt to make this intermediate indolenium, and this could be quenched internally via cyclization to make these type of proloindolene. And so it's important to note, number one, that there's no palladium in this reaction anymore, and more importantly, that the enantioselectivity of this initial amination reaction would be determined by this chiral anion. And so what we did is we took this protected tryptamine and tried with these conditions. We're able to isolate out this C3 diazonated pyrroloindolene in pretty good or very good yield and some reasonable levels of enantioselectivity. Shown here on the right is an ORTEP diagram, which confirms unambiguously that these are the types of molecules that we were synthesizing. And so it turns out, I think, another example of how we set out to, prove our methodological hypothesis, and that we ended up making complex molecules. So this type of C3 diazonated pyrroloindolene has been proven by Movasagi to be a very useful intermediate within the context of total synthesis, complex molecule total synthesis. So he's taken this tryptamine derivative, and in a 5-step sequence, 5-step diastereoselective sequence, made this type of C3 diazonated pyrroloindolene. And furthermore, you can take these under photolytic conditions and extrude N2 through a fragmentation process and recombination to make this type of C3 arylated pyrroloindolene with good levels of enantioselectivity. And so we spent some time optimizing this process. We're able to take these benzamide protected pyrroloindolines, using this spirotrip catalyst shown here on the bottom right-hand corner. We're able to make these diazines in up to 91% E, 99% yield. And furthermore, we can photolyze these compounds to make the C3 arylated compound, the C3 arylated products. And so here's a picture of these reactions. So these C3 diazonated prolyndolines are bright yellow. And so it's actually very easy to run these reactions in the laboratory and get a rough idea for what works and what doesn't work. So usually when they're done, they're this bright yellow color. When they start, they're colorless, and when they go bad, I guess it's kind of cut off in this picture. When they go bad, they're this red color in the back. And so it's really easy to run a lot of these reactions. We're able to optimize this process in very short order, in 2 or 3 months. And so we have a lot of different examples where this works. We can substitute our aryl diazonium cations. We can substitute the indole moiety. We can substitute the protecting group on the tryptamine. We can substitute the indole nitrogen as well, and still have reasonable to excellent levels of enantioselectivity selectivity and really good yields. So we wanted to take this process. So I guess in some respect, we kind of stumbled across an enantioselective electrophilic amination. And so the first thing that comes to mind within the context of that type of reactivity is, can we make amino acids? Those are useful for human health, catalysis, a lot of reasons. And so what we envisioned is that, or what we set out to do, was take beta keto esters such as this indinone shown on the left, and under our very identical conditions to the prolyndolene chemistry, make alpha diazonated amino acid type compounds. And so initial efforts were able to achieve 32% E, and then switching base from sodium phosphate to this dibasic sodium phosphate or monobasic sodium phosphate led to a huge increase in enantioselectivity. And so what we hypothesized was that sodium phosphate's too strong of a base, and so we want to make sure that this indinone sits in its enol tautomer form, and so sodium phosphate would just deprotonate that. And with this weaker base, what happens is we can get two interactions. Basically, our phase transfer catalyst can tether our enol nucleophile to our aryl diazonium cation, and you get a highly ordered transition state. In this case, we're able to achieve enantioselectivity, and in fact, we think the base is just there to go sweep this proton off of the oxocarbenium initial product. And so we're able to use this to make a lot of different compounds. We can substitute the indinone aryl ring, the aryl diazonium aryl ring. We can put in different protecting groups, so we can use beta ketoamides. We can use different size rings. So this benzosuberone works very well. And we can also use enamine type nucleophiles and in that case, don't have to have an alpha withdrawing group. Excuse me. And so the products of these reactions are fairly useful. They're amino acids. And so we can take the diazine and reduce it under standard conditions to get to these types of amino alcohols. We can do a further reduction using palladium chloride and polyhydroxymethylsilane and methanol solvent to get to this fully reduced compound. And this compound's pretty interesting. So if you look at this structure, it's actually a conformationally constrained tyrosine analog. So it's basically tyrosine that's tied up. And so people are interested in using these compounds for a variety of applications. And usually people prepare this compound in 12 steps, and they enantioenrich it via classical resolution. And so we're able to achieve this using very low catalyst loading, so a couple mole percent of chiral material, and in a 3-step process. We're also pretty excited that we can use this methodology to do enantioselective N15 labeling. So some people may want to use this for protein structural studies for NMR and things like that. And so we're able to use relatively inexpensive sodium nitrite, and in a 3-step sequence get to this enantio-enriched, N15-enriched, conformationally constrained tyrosine analog. And finally, one of the cooler examples from this project, which is still under development, we have a postdoc currently in the TOS group that's working on this, is we can do macrocyclizations enantioselectively. And so in this case, we're able to take this aryl diazonium salt with a tethered dicarbonyl nucleophile, and using our phase transfer strategy, get to the 16-membered ring and 46% E. And so what's unusual about this, at least from my perspective, is that usually macrocyclizations like this, you do at very low concentration, so 0.0001 molar, because you're afraid of oligomerization or dimerization, and you use concentration to control that. But here, forgetting about the enantioselectivity, we're using a phase transfer process to control macrocyclization. So basically, we're only letting very small amounts of our substrate into solution where it reacts. And so hopefully we can develop this chemistry, and hopefully you guys will be able to read about it at some point in the near future. Okay, so I kind of ran through a lot of chemistry today, and especially in the second part of the talk. And so basically what I told you guys about is how we could use a chiral anion phase transfer process, specifically of aryl diazonium salts, to do a number of reactions. We can do borylation, um, uh, annulation reactions, arylation reactions. We can do an antioselective amination on a number of substrates, and even an antioselective macrocyclization. So the first part of the talk, I told you about work that I did during my PhD. The second part, I told you about work that I did during my postdoc with Dean Toss, UC Berkeley. So this is work that I obviously did in my previous groups, but there's a lot of people there that need to be thanked, specifically Professor Brian Stoltz and Professor Dean Toss, who were my advisors on these projects. The Stoltz group in total, the Toss group in total, Sarah Reisman's group, Richmond Sarpong's group, also were next door neighbors and very helpful with reagents. I had a number of project mates: Dr. Johnny Gordon, Dr. Kamer Akami, Dr. Scott Virgil, Dr. Brett Williams, Jigar Patel, Dr. Hunter Shenantoa, Javier Arenas, Tao Wu, Gajendra Ingle, Saul Reesberg, Linda Choi, Joshua Owen, Angel Alexiev, and all of this wouldn't be possible without generous funding from the United Negro College Fund, the Department of Energy, Merck, Ford Foundation, NSF, and both of these wonderful institutions that I just told you about. And before I finish up, I want to give a bit of a sales pitch for UCLA. And so it's a beautiful place. You can see pictures of the campus. We have wonderful sunsets. It's a unique situation. So we have a lot of trees, and it feels, at least the area immediately surrounding UCLA is very not rural, but it has a very outdoorsy feel for being in such close proximity to such a major, huge city today. And so this is a slide that I've never changed, and I can just say it's 80 degrees Fahrenheit today, because that's true almost all the time. And so here's a graph showing how wonderful the weather here is. So these are the lows down here. It never ever gets below freezing. We're situated in a very interesting area of Southern California and Los Angeles. We're between Bel Air, Santa Monica, Beverly Hills, and Hollywood. And so these are, if you're into star watching, there's no better place in the world to be. And so there's a lot of stuff to do around here. So we have wonderful beaches that you can go to year round. The nightlife in downtown LA is also very good and very up and coming. There's a lot to do around the city. And the campus is beautiful. This is where I work. This is molecular sciences, and my office is right around there where I'm sitting right now. Young Hall is also a chemistry building. Boyer Hall is where most of the biochemistry is done. And so we have a huge department, 55 professors in chemistry and biochemistry, 1,400 undergraduate students, 350 PhD students, and 100 postdocs. From that, there's some Nobel laureates walking around, Presidential National Medal of Science recipients, quite a few members of the National Academy, all in our department and active researchers. And I think I guess I'll leave you saying UCLA was ranked recently as the number 5 university in the world overall for chemistry. And so with that, I'll be happy to take any questions about my research. And I thank you guys all for your attention.
Okay, thank you very much. We have a question. Remember to say your name.
Hello, my name is Emery Dent. I'm a biochemistry major here. And in reference to your doctoral studies, you're talking about, just got to get terminology here, transagonalides and vacilliolides, and basically synthesizing them to get some sort of base that's similar, identical to the natural compound, correct?
Yeah, so I mean, the thing is, the compounds we synthesize are spectroscopically, you cannot differentiate them from the naturally occurring compounds. So
Okay.
we synthesize the compounds, right? So
Yeah. So my question is like, after all of the time and labor and I guess, chemical costs and stuff like that, like, um number one, I just to clarify, is it possible to just pull it naturally? And number two, what is the comparison after
Yeah,
all of that is?
absolutely. So it's a great question. And so the first thing is that, okay, so if you read medicinal chemistry, what you'll find out is that around 3/4 of the drugs that exist are based on natural products. And the reason why I say based is because a lot of times they're derivatives. And so you'll have some basal biological activity in a naturally occurring compound, and then you can tune it. It's pharmacokinetics. You can tune a lot of things by changing the structures. And so the thing is, by developing a total synthesis of a bioactive compound, I can do, or we can do, or some, whoever's researching can do whatever they want to change that compound structurally to tune it to their application. That's the first part of it. So it's really important that it's modular. We can make derivatives, essentially. The second thing is, for example, in this case, it's very true. So transagonalide C, for example, is a SERCA inhibitor, at least proposed at that time to be a SERCA inhibitor. However, transagonalide C comes from the same plant as thapsigargan, one of the most potent SERCA inhibitors known. And so there was a lot of debate whether the SERCA inhibition that was observed from transagonalide C was just a result of contamination. And so we gave synthetic transagonalide C to biologists who were able to actually measure that and say, no, this compound's also a SERC inhibitor. It's not a result of contamination from another compound. And so in terms of synthesizing these naturally occurring compounds, there's certainly a tremendous advantage in terms of application. And that, in addition to that, the people that we train, so I mean, it's you mentioned the cost, but all of the chemists that work in pharmaceutical companies, all of the chemists that work at the FDA, I mean, the whole machinery, all these people have to be trained. And so total synthesis is certainly a fantastic training resource, the way that we can educate synthetic chemists, because it really represents some of the biggest synthetic challenges, right? Nature is a really good synthetic chemist, and mimicking them takes a lot of skill. Does that answer your question? Yeah, I guess so.
My name's Emily, and I'm wondering what the difference in the Ireland Claisen that you talked about is compared to a regular Claisen.
Yeah, so there's a lot of difference, and so I didn't have time to talk about it. But the Claisen rearrangement that I told you, in what's different, I guess I'll flip to the slide. It'll probably be easier to see. Actually, I have a backup slide, probably specifically to this topic, because at some point it was in my talk. Okay. Okay, getting there. Okay, so the Claisen rearrangement. Okay, so there's the biggest feature of the Claisen rearrangement, what makes it a famous classic reaction, is that you can get a highly diastereoselective reaction if you can control ketene acetal geometry. So basically, whether this pyrone, if you can see where my cursor is going, is in the pseudo-equatorial position or pseudo-axial position. And depending on which way it stands, you can get the Z ketene acetal or the E ketene acetal. So it's very famous for that. That's the first part of it. And so what happens in systems like this, the one that I'm using, where you have this electron withdrawing group. So this is electron withdrawing group. And an electron withdrawing group alpha to this carbonyl makes it really difficult to form the E or the Z geometry, right? Because it isomerizes. It's configurationally unstable. And so that makes this system unusual. So you don't find many examples of people using esters with alpha withdrawing substituents to do Claisen rearrangement. That's the first part of it. And I guess the second part of it is that actually the product of reactions, or the product of Ireland Claisen rearrangements that have this withdrawing group, they're actually prone to undergo decarboxylation. And so there's a reaction called the decarboxylative Claisen rearrangement, which is essentially a substrate just like mine, and it has a withdrawing group. It just happens to be a little bit more withdrawing. And so when it forms that immediate Claisen product, it just loses CO2. And there's another rearrangement called the Carroll rearrangement. And you can look those two reactions up and see that those are essentially both just Claisen rearrangements followed by decarboxylation. And so it's challenging in that regard. And that's what makes them different, is that we use these types of substrates.
Hi, my name's Gabriel. I have a question about your PhD research.
Yes.
I saw that you had some really low yields in your
Yeah.
synthesis. I think
Yeah.
one was as low as 35%.
Mhm.
From what I know about organic synthesis, that's a terrible yield. How does
Yes.
that affect a multi-step synthesis?
So the thing is, so it's a balance, right? And so I guess I'll try and find that slide. So so I'll say a couple things. So the first thing is that someone else made this molecule and they had greater than probably 80% yield on almost all the steps, but they did it in 30 steps, right? And so that's the reason why, at least in this case, we found it acceptable to have such a low yield. Because essentially, so this step is 31%, and that's a mixture of two diastereomers. I mean, both diastereomers are natural products, so it's okay. But we can do this in 7 steps from commercial. And so, furthermore, I've had undergraduate researchers who are green able to make this intermediate in 2 weeks. And so, what that means is, yeah, the yield on this last step is not great. But if you've ever done a 30-step synthesis, even if you're an experienced chemist, that takes a lot of time, months, right? And so this is a 2-week process from beginning to end. And so it was something that we had to deal with. And so I'll note that. So in this other synthesis I'm citing, I don't have it written here. They have an intermediate very similar to this, but they use I think it's a 6 or 7 step sequence to get to the natural products. And so it's low yielding, but it is the most direct way that I can envision. And so, yeah, it's a compromise. I would have loved to see this reaction in higher yield. And so we have, in a later publication that I didn't have a chance to talk about today, we did spend some time optimizing the yield of that reaction.
Hi, my name is Adriana, and about the Thaspia plant, what kind of property made it able to treat blemishes or bruises, but made it so toxic when ingested?
Yes, that's a great question, and I actually do not know the answer, and I'm very glad that I've been stumped finally on this. Yeah, so it's interesting, and so if you look in the literature, it is both an irritant and capable of treating irritation. And that sums up how its bioactivity is described. I don't know. It's a great question. I don't know if it, for example, is so deadly to cells, it just goes and kills all that layer of skin, and then your blemish is gone because the skin that it was blemishing is no longer alive. And so, yeah, I don't know. That's a great question, and I should probably know that. Thank you.
Hello, my name's Noah, and I noticed that there were a lot of times, especially during the first subject you were talking about, that you had to do a lot of minute adjustments each time. Were there ever any moments where you kind of just wanted to give up, move on to something else?
Yeah, so the thing is, okay, so this is a 5 years of work, and not a little bit of work. People in my field, in my discipline, work very hard in terms of hour and labors, and so the amount of adjustments that you saw was actually insignificant compared to the actual amount of minor adjustments that were made. So yeah, I mean, at times it's frustrating, but I think the end result and the take home message for someone who's aspiring as a scientist, that if you want to quit, it may not be for you. I mean, if you're not driven, you have to, it's like you really have to be driven to succeed in this game, and especially in total synthesis. And so, yeah, there was not necessarily points I wanted to give up. There were points that I was discouraged, but my overarching scientific desire or interest in finishing the project prevented me from giving up, if that makes any sense. I mean, there was a lot of painful time, absolutely, but in the end, we still wanted to make the compound, and that was greater than all other challenges. Thank you.
Okay. So one last question, and I think I'll ask the last question. What targets are you getting at now with your, starting up your research in your own lab?
Yeah, absolutely. So
Yeah.
I don't want to talk about that because someone's going to steal it. No, so the thing is, so we're doing, actually a lot of the chemistry I'm doing is very similar to my training and a natural offshoot. So I'm doing some natural product synthesis. The compounds I'm targeting are actually a bit smaller than these compounds, and so what I found is actually relates to the last question. Is total synthesis can be a very discouraging art, and for me, I've tried to select targets for my students that are a little bit smaller, more manageable, can be completed in a shorter amount of time, and also have perhaps more applications even. And so there are some natural products that I'm targeting. Specifically, I'm interested in compounds that are perhaps useful for neglected diseases, and also antibiotics.
Good. That's good enough.
Yes, got to help people, right? Yeah.
Yes.
Yeah.
All right. We want to thank you again.
Thank you, guys.
Okay.